You have about 6 seconds.
I think it might have been a clicker. I'll just get started, shall I? So, morning everybody. I am Tyler Larsen from the Fowler-Strausman Lab at Washington University in St. Louis. And I am interested in studying how different species interact and how those interactions are involved. And so I won't have to tell anyone in this room here that species interact all the time, and they interact in lots and lots of different ways. And one important way that we can classify species interactions is based on the fitness consequences on the partner. So we can imagine we have mutualisms where multiple participants are benefiting, or various antagonistic ones where one participant is benefiting at the expense of the other. But of course, it's more complicated than this. Oftentimes, interactions involve both positive and negative consequences for the participants. And also, these interactions can move due to changing environmental context or evolution of more or less antagonism. And so my research is interested in asking how and why do interspecies interactions evolve to become more antagonistic or less antagonistic. And in pursuit of answering some of these questions, I am using two, I would say, very interesting microbes, Dictyostelium discoideum and bacterium in the genus Burkholderia. So Dicty, if you're not familiar with it, is an extremely cool organism. I don't have time to go into all the great reasons that it's such a cool microbe. But if you don't know, look at it on Wikipedia. But in short, it's a social amoeba. It spends most of its time as a single cell living in the soil, but then can enter this very bizarre social life cycle where they come together and form these multicellular structures and do a bunch of cool things. Lots of neat biology, lots of interesting things have been studied using Dicty as a unique model system. But for my purposes today, the important aspect of Dicty is that many Dicty isolates taken from the wild carry these bacterial endosymbionts from the genus Burkholderia. There's a couple different species. So you can see on the right there, there's a micrograph showing the purple ones on the Burkholderia inside of Dictyostelium spores. And so these are bacteria. They infect intracellular, they get phagocytosis and somehow don't get digested, and they can persist and hang around with Dictyostelium for a long time. And what's cool about their interaction to me, there's lots of great reasons to study these two organisms, but for me, part of it is that they have this very complicated interaction with both positive and negative consequences for both of these species. And so ultimately, I'm interested in trying to tease some of this apart. And so for my talk today, basically I'm trying to ask, are they friends or foes? And while obviously I'm a fan of these species and there's lots of great things about studying them, one important limitation is that while they're easy to study in the laboratory, they're very difficult to study in nature because they're kind of small. And it's difficult to actually capture the specific ecological context in which this interaction occurs. And so to try and answer if these guys are friends or foes, I'm taking this very roundabout, indirect, experimental evolution sort of tactic, where in short, I am trying to infer the existence of adaptations by evolving them back away. And so the central logic here is that if Burkholderia and Dicty exert some sort of selective pressure on each other and have historically in the wild, if I separate them apart and evolve them in isolation in the laboratory, we should see those many adaptations that arose as a result of those selection pressures should tend to disappear, just due to genetic drift or adaptive streamlining. And so that's basically what I did. I took a great many different strains of Burkholderia and a great many strains of Dictyostelium, separated them out, evolved them in the lab with no access to their partner, and then at the end I can put them back together in various combinations and try and see how has their antagonism with local population changed. And so I think a cool aspect of this approach is that it should work whether Dicty and Burke are friends or foes. You can imagine them losing adaptations that made them friendly or losing adaptations that made them antagonistic. And so I did a whole bunch of different assays to try and see how this antagonism or cooperation may have changed. They don't bear going into great detail right now. I'm sorry. But the first one, the gist of it is I'm looking at dictyostelium's fitness when it's exposed to Burkholderia. So Burkholderia, when it infects dicty, it's somewhat toxic. And so dicty produces fewer spores, it's less fit when it has Burkholderia inside of it, for the most part. And so I can basically do this assay to look for how this toxic effect may have changed when I evolved Burkholderia by itself, or how dicty's susceptibility to this toxicity may change when I evolved dicty by itself. So all my data are going to look kind of like this. It's less visible than I was hoping. So imagine, if you will, there are 6 gray boxes. They're really light gray. And the 6 gray boxes, oops, 1, 2, 3, correspond to different strain pairs of dicty and Burkholderia. Those are ones that we isolated together in nature, so we know they interact in nature at least sometimes. And in each of these facets, the red bar represents the ancestor dicty and the ancestor Burkholderia paired together, and the blue bars represent evolved of one partner, 3 different evolved lines of one partner paired with the ancestor. And so, for instance, we can see in the last facet, QS69, basically we're seeing Burkholderia become more toxic when I evolve Burkholderia in isolation. So it's not, the horizontal black line represents 0 toxicity, and so the red bar is at the far right, it's up there close to 0. And then the evolved lines are more toxic. So by the logic I described earlier, if they're becoming more toxic when they don't even have access to dicty, they may have had adaptations that made them less toxic in the first place. So this may be evidence for non-antagonistic or more cooperative elements to their relationship in some of these strains. Similarly, if we look at how dicty changes, this is again the susceptibility of dicty to this Burkholderia toxicity. We can see that generally, several different strains, we've got dicty becoming more susceptible to Burkholderia's toxicity, which kind of contradicts the first result, suggesting that dicty has some mechanisms to resist this toxicity in the first place. A second assay, conceptually similar, is basically looking at dicty's toxicity to Burkholderia. So dicty has a growth suppressing effect on Burkholderia, and I can look at how this evolves and how Burkholderia's susceptibility to it evolves. And in short, we see that several strains of dictyostelium become less toxic, so they seem to be losing this growth suppression trait, and then several Burkholderia strains became more susceptible to it, so they're losing some sort of resistance to it. And then the third assay I'm going to bring up briefly is using qPCR to estimate how many Burkholderia we end up inside of each dictyostelium spore, or cell rather. And so this is trying to capture both sort of how suitable a host dicty is for Burkholderia, but also how effective Burkholderia is at persisting and reproducing inside of the dicty cells. And what we see is that some Burkholderia strains become less infective. We get fewer total Burkholderia inside of each dicty cell when we evolve Burkholderia. And when we evolve the dicty, some of the dicty strains become more susceptible to infection and have higher loads of Burkholderia in them over time. So I've got a lot of results, and I'm trying to understand them all and put them together. But what can I conclude so far? Basically, I do see changes in both species when I evolve them in isolation. And if my logic holds, this may be evidence that their interaction does happen in nature and does exert some sort of selective pressure on the 2 species involved. Most of my results point in the direction of that they're sort of antagonistic in general in nature. But I didn't have time to mention that we have a lot of interesting laboratory evidence that it's complicated. So there's more to tell in this story. But it's also clearly true that there's a whole lot of strain-to-strain variability in the results of that. And so if you're asking where does the interaction between Dicty and Burkholderia land on this antagonism to cooperation spectrum, the answer seems to be sort of smeared all over the middle. And so, who cares? Well, I do, but I hope I can convince you to do a little bit. Ultimately, this is in service to, I want to develop Dicty and Burkholderia into a useful system for actually doing experimental coevolution, as this session's name, the term didn't come up. Evolving them in the same dish instead of separating them apart and seeing how they become less natural. And so I'm in the progress working on an experiment where I evolve the two of them together under various conditions that theory predicts should lead to more antagonism or less antagonism. Yes, specific hypotheses. And so I'm pretty excited about that. And I'm also doing sequencing with a bunch of other hypotheses. So I hope to see you guys next time, and I'll have more to say. With that, I'm happy to take any questions. I practiced this like 10 times, and I never got under about 20 minutes.
Have you thought of, I love your talk, have you thought of, or are you already looking at the nutrient environment of the slime? Low nutrient environment versus high nutrient environment, because that might affect the outcome of it.
Yeah. So one thing that I didn't really talk about, and if you'll recall in that first micrograph I showed of Burkholderia inside of the Dicty cells, there was another bacteria in there too. Burkholderia is not really edible to Dicty. And so there's at least sort of the big interesting thing that Burkholderia infection does for Dicty, the potential benefit of infection, is that when infected with Burkholderia, Dicty can carry other species of bacteria through its social cycle, sort of pack a lunch. And so there's lots of interesting stuff that our lab is looking at along that line. But I think what it definitely tells us is that the biotic context is going to matter a lot. And we have seen that the number of bacteria that we plate for each Dicty, not only with the food bacteria, but with Burkholderia, can have a big effect on the assays that we do in the lab. And that's part of why I kind of like this approach that I've taken, is because while we've done a lot of cool stuff with a lot of neat assays, we don't know how many bacteria there are in the wild for each Dicty, for instance. That's one of those sort of ecological questions I think is very difficult for us to get at. And so in designing this study so far, I've tried to come up with a way that it maybe doesn't matter if the assays directly replicate the biology of nature, so long as we're seeing how the assay changes over time and sort of further back on that. I don't know exactly. Anyway, it matters a lot, and we're all looking at it.
How quickly were you seeing the antagonism or lack of antagonism evolve? I don't know if it's how many generations.
So it's a lot more generations in the Burkholderia than the Dicty. We've made some estimates. Short answer is that when I did the experiment, it was 2 months. So it was 30 transfers in 2 days. 2 days apart. And that, I think, mostly proved to be sufficient. Obviously, more would be better. But for future experiments, I'm planning to do about the same period. So, dozens of generations. We can talk about that. Thank you.